Good afternoon, welcome. My name is James. I'm one of the house managers here at Town Hall. On behalf of the staff at Town Hall Seattle and UW Engage Science, it's my pleasure to welcome you to the first of 5 in our series with some of the students who are leading the latest wave of scientific discovery in our region. Today's presenters are Megan Gialluca, Abby Elerding, and Emma Scalisi. As we get underway, I would like to acknowledge our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continuing use of the natural resources of their ancestral homeland. We're so glad to have you join us today. The presentation will run about 90 minutes, including the Q&A, at which point this microphone will be down in the corner there, and you can come up. We ask that you keep your questions concise as possible so we can get to as many as possible. Town Hall is adding new events and podcasts every day. Our next science event will be on April 28th with Sandy Sabursky and Mary Grace Becker. They will be providing resources and meaningful connections for people with dementia, as well as a way for care partners to grow. We will also have another UW Engage Science event on May 3rd with more speakers and more new research. Visit our website to join our email list and get the latest updates as more programs are added throughout the season. Town Hall's work is made possible through your support and the support of our sponsors. As part of our Arno G. Matulski Science Lecture Series, this event is sponsored by Microsoft. Town Hall is also a member-supported organization, so I'd like to thank all of our members who are joining us. If you share in Town Hall's goal of a community energized and empowered by considering questions of politics, science, and culture, please consider supporting us by becoming a member. You will find membership information on our website. And now join me in welcoming UW graduate student and instructor of the Engage Science Communication course this year, Nicole Gregoria, to introduce the program and our first speaker.
All right. Hi, everyone. Thank you so much for being here tonight. Last winter, I was very honored to get to instruct our Engage Science Communication course. This course, along with other workshops and events, are run by a group of graduate students at the University of Washington called Engage. The course itself seeks to prepare a group of excellent UW graduate students with the skills they need to communicate about their work with the general audience. Along with our other efforts, the Engage group hopes to improve science communication education at the University of Washington, and we envision a future where scientists more effectively and freely communicate about their work with the public. Tonight, 3 of our incredible students from the course are here to tell you about their graduate work. Their talks represent real research happening at UW right now, so we hope you enjoy this unique opportunity to get a behind-the-scenes look at it. Without further ado, I'd like to introduce our first speaker, Megan Gialluca, who studies massive water loss on planets in other solar systems called exoplanets. This process can turn a potentially habitable planet like Earth into a burning, hot, waterless environment like Venus. Understanding whether or not a planet has undergone this process informs scientists on where to search for alien life in the universe and what the clues we should look for are. So please welcome Megan.
Okay, nobody look at my slides while I pull them up. Great. All right, hello. So when I was in high school, I took an AP US language class. And our final for that class was to write an argumentative, persuasive essay. And at the very bottom of that assignment, it said, but don't choose any silly topics. For example, why are aliens real? And I think you know what happened next. From that essay was born some of my favorite quotes of myself that I've ever said, including, the first argument I hear is we don't have proof that aliens exist. Well, we don't have proof that they don't. And if you want to think about it in technical terms, we are aliens. And my personal favorite, not only do I think this, but some of the biggest names in science do. And so, since who you are in high school is completely indicative of who you're going to be when you grow up, it might not surprise you to find out that I went into the field of astrobiology. And now, astrobiology is not the belief that we've been visited by little green men, or even that that could happen in the future. Astrobiology is simply the study of life in the universe. And that life can include things like bacteria, like plants, and all sorts of things. It's also the study of the origin of life on Earth. Now, before I get to the main talk, I'm going to answer some of your dying questions for astrobiology, because everybody has these. NASA is not hiding any secret aliens that I know of. I don't think we're part of an alien experiment. An exoplanet is a planet that in a different solar system, so around any star that's not our sun. Technically, I'm a Gemini. And perhaps the most important question of all, aliens can be any life form born off of Earth, including plants and bacteria. So if you go outside tonight and look up at Mars and see a tree growing on the surface, congratulations, you've just discovered the first alien life, and you will undoubtedly get the Nobel Prize. So that brings me to the primary subject that I'd like to talk to you guys about today, which is how to search for life in the universe. And now I am an astronomer by training. Astrobiologists can be from any sort of scientific study, including chemistry and geology and biology. But I'm an astronomer, so I'm going to give you the atmosphere edition. And it'll make sense why that is, hopefully, to come. So imagine you're an alien on another planet, and you're looking at Earth, and you want to prove to all of your other little alien friends that there is life on Earth. How would you go about doing it if you can't actually see the surface? Well, there's 3 primary ways you could do it. Number 1 is plants. You could look for the plants on our planet. Number 2 is technology, or what you might hear called technosignatures. That's like contact with Jodie Foster. And then the 3rd way that you can search for life on another planet is in the atmosphere. And as I already said, I'm going to focus on that. So the atmosphere is maybe less intuitive than these other methods. And so I want to talk to you about why we can look at an atmosphere, which is the air all around us here on Earth that you are hopefully breathing right now, and tell from that air that there is life on the planet. And to do this, let's talk about Earth's entire history in just a few minutes. I am going to tell you how life has changed the atmosphere and why we could prove from those changes that life exists. So this starts 4.5 billion years ago in the Hadean Eon. This is when the Earth was formed. This is when the whole solar system was formed. So at this time, Earth is just a big ball of molten rock and lava. This is also when we have the moon formation. And then at the very end of the Hadean Eon, and we don't know exactly when, exactly why, or exactly where or how this happened, but life started here on Earth. And we know that it happened then because in the Archean Eon, which is the next eon in Earth's history, we see the first evidence that life was on Earth and it was growing and surviving and thriving. And that evidence comes in the form of a 3.7 billion year old fossil called a stromatolite. So at this time, the very earliest types of life on Earth were these things called methanogens. So they probably looked like that, but I don't know. I'm not a biologist. And these methanogens just like to spit out methane. They breathed out lots of methane. And they would live together in these little communities, and their excrement would build these rock formations, which are these stromatolites. And that's, of course, our evidence that life existed at that time. But what's really important is that these bacteria were breathing out methane. I want you to remember this because that methane goes into the atmosphere. After the Archean comes the Proterozoic Eon, and this eon is super important. It's marked by something we call the Great Oxygenation Event. So at some time towards the end of the Archean, we have photosynthesis develop. And nobody knows exactly which player had the biggest role in this. It was probably cyanobacteria or some other kind of bacteria. Maybe it was plants. But these things started photosynthesizing and producing oxygen. And they flooded the atmosphere with oxygen, so much so that it increased by 200 to 400 times the levels of oxygen that were in the Archean. And at this time, there's lots of other stuff going on in the Proterozoic, such as the ice ages, and then the Proterozoic ends and the Phanerozoic begins, which is what we're in today. The Phanerozoic begins with something called the Cambrian explosion. This is the explosion of large multicellular life on Earth, like the trilobites, like all of these guys you see in the picture, the dinosaurs at some point in the Phanerozoic, and then all the way at the end, towards present day, of course, we have humans. So I've just given you the entire history of life on Earth, and we've done it from maybe a historical, biological, geological perspective. Now I want you to put yourself in the shoes of an astronomer and think about how you might watch life evolve on Earth. And it would probably look something like this. We have the no life period, and then the life produces methane period, and then finally the life produces oxygen period. And at all of these times in Earth's history, the air on Earth, the atmosphere, is going to look different. And you're going to find different things in it. And so in the no-life period, maybe we have nitrogen. That's kind of just this chill gas that likes to hang around. Our atmosphere currently is mostly nitrogen. And then we have lots of methane that we could see, and then we have lots of oxygen. For those last two gases, if we were to observe those in Earth's atmosphere, well, life produced those gases. And if we detect one of those gases, we've detected evidence of life on Earth. So if we detect a gas produced by life on another planet, we call that a biosignature, or a signature of biology. And there's lots of biosignatures we can look for. Some of the most common ones are ones we've already talked about, methane, which is produced by bacteria, and also nowadays it's produced by big life, like cows in industrialized agriculture. There's also oxygen produced by photosynthesis. Ozone is a new one. It's basically oxygen. The oxygen you breathe is 2 O's. Ozone is 3 O's. And so it's just created when there's already a lot of oxygen hanging around. And then there's also nitrous oxide, which is primarily produced by bacteria in the soil during something called the nitrogen cycle. And so at this point, I've told you these biosignature gases are produced by life. So we can see them in the atmosphere. And you're probably thinking, awesome, if we detect one of these, we've detected aliens, right? Right? Not quite. It's never that easy, right? And unfortunately, we can't trust these biosignatures because although they are all produced by life, they are also produced by not life. So methane is also produced by volcanoes and metamorphism when rocks are heated and crushed. Oxygen is also produced by sunburnt water or highly irradiated water, which I'm going to talk a lot more about. Ozone, since it's just oxygen, it can also be created by sunburnt water. And nitrous oxide is created by lightning. So let's take a closer look at oxygen. And I'm going to hopefully give you a glimpse into just how difficult it can be to figure out if something is life or not. So first of all, why oxygen? Oxygen is always talked about as being akin to life. Life needs oxygen. Life produces oxygen. It's the king of all biosignatures. But nowadays, we're kind of finding out that this isn't true. And this is what I specifically research at the University of Washington. So oxygen doesn't like to be by itself. It won't just hang out in the atmosphere. It likes to leave and become a part of other things. Two of those things could be volcanic gases. It'll form carbon dioxide. It will also be involved in surface weathering, so what you might think of as rusting. And in order to have oxygen hang around in the atmosphere in substantial amounts, you have to be constantly producing it and constantly flooding the atmosphere with oxygen as these processes desperately try to remove it. And life does that really well, especially here on Earth. And now we have an atmosphere that is nice 21% oxygen, and the rest is basically nitrogen, but 21% oxygen, that's pretty good. That's a lot of oxygen. Unfortunately, you can have just this much oxygen through a whole other process that I'm going to describe to you. And this process is specifically what I research and what I model and try to understand. So on the left, I want you to imagine you have an atmosphere, and that atmosphere is just filled with water. And now water is everywhere in the universe. It's everywhere. Every planet probably has some amount of water trapped somewhere, whether it's in its interior, whether it's in ice. It's everywhere. And then we also have a milkshake. And the milkshake is mint chocolate chip because it's my milkshake, my choice. And I want you to think about the ice cream in that milkshake, the mint ice cream, as being hydrogen. the chocolate chips as being oxygen, and so when you have it all together, you have water molecules. And just like our milkshake, the atmosphere can split these water molecules apart, so it can have just hydrogen and just oxygen hanging around too. And now, in the case of the milkshake, you or your lips are going to be the thing that is causing the ice cream to come out of the cup, causing it to leave. In the same way, the sun or the star of that planet is going to be the thing causing the water and the hydrogen and the oxygen to leave the atmosphere. And you can think about why that is as like a rolling boil of water on your stove. It kind of evaporates that water up out of the atmosphere. But anyways, as time goes on, you're going to start drinking your milkshake, and you're going to be pulling hydrogen or ice cream and chocolate chips, and the sun is going to be pulling water and oxygen and hydrogen. And it's a little bit easier to pull ice cream through a straw than it is chocolate chips, so you're going to get more of that. Just like it's a little bit easier to pull hydrogen out of the atmosphere than it is oxygen, because oxygen is heavier. And as time goes on, you're going to be plagued with just a god-awful issue, which is that the chocolate chips are going to get stuck in the straw. And as soon as that happens, it becomes very difficult to pull any more chocolate chips through the straw, because every time they go up, they're just going to get stuck by that blockage. And the same problem is going to happen in the atmosphere. Some oxygen is going to get stuck, and it's going to cause all the rest of that oxygen to get stuck as the water is trying to leave. But you can still pull ice cream around those chocolate chips, and you can still pull hydrogen around the oxygen. So you just keep going, and you keep drinking, until you're left with just a pile of chocolate chips in the bottom of your cup because you've drinking all of the ice cream. And in the atmosphere, you're going to be just left with a big pile of oxygen because you've taken out all of the hydrogen. And for you, that's not really a big deal because you'll just toss your cup away, no problem. But for a planet, this is a really, really big problem because once you lose your hydrogen, you also lose the ability to create water molecules. So not only do you have an atmosphere that's filled with oxygen, you also have no water. And that means no life as we know it here on Earth. And this isn't just some like crazy theoretical process that I came up with to prove to everybody that oxygen doesn't work. This actually happened in our solar system on Venus, way back in the early solar system. And nowadays, Venus is completely devoid of water, and it's 900 degrees Fahrenheit on the surface, which is not good if you want to live there. And so this is the process that we could have a planet create lots of oxygen with no photosynthesis necessary. And so just by looking at that oxygen, we have no way of knowing if life created it or if it's what we call a false positive or a fake biosignature. So you have one scenario where plants create it, and that's great, it's a biosignature. The other scenario, water and radiation create it, and it's a false positive. And our detectors have no way of telling the difference. If you point a telescope at that planet, all it's going to tell you is, yeah, there's oxygen there. Good job. So that doesn't really work for us when we want to say beyond a reasonable doubt whether or not life lives there. But luckily, we can get creative. So one strategy is to use lots of biosignature gases. You'll remember in the beginning or towards the beginning, I talked about some of the most common biosignatures that we look for. Well, maybe if you just see oxygen, you're unsure. You're not really sure if life created that or not. But what if you detect oxygen and methane? Well, all of a sudden, the probability that life is on that planet gets a little bit higher because it gets a little bit harder to explain both of those gases being present without life. Another strategy we can use is to use more than just the atmosphere. You'll remember at the very beginning, I talked about lots of different ways we can prove there's life on the surface. So maybe you can't just look at oxygen, but what if you detect oxygen and you have pretty strong evidence that there's plants on the surface too? Well, all of a sudden, it gets a little bit more likely that that's life on the surface, since plants produce oxygen. But the bottom line is, we have to work together. There's lots of different branches of science that work together on this problem of finding life in the universe. It's not just astronomy, and it's not just biology. I know nothing about plants. So I need a biologist to tell me about that. And then I need an atmospheric scientist to tell me how long those produced gases will live in the atmosphere. And then me as an astronomer might tell you, yes, you could detect that, or no, we can't detect that. We should probably find a different target. And the future is bright, literally. The joke is that telescopes look at light, by the way. So at present, in December of 2021, we had the JWST launch. And so that telescope was a massive effort. A lot of people kind of refer to it as Hubble's replacement. And JWST is now currently looking at planets, and it's looking at all sorts of stuff in astronomy, but what I'm interested in are the planets. And it has already observed a couple of our most promising astrobiological targets. And what's even more exciting than JWST, can you imagine, is the Habitable Worlds Observatory. Every 10 years, NASA conducts something called the Decadal Survey, where they basically say, okay, this initiative, this is where we're going to put all of our money because it's the most important thing. And the Habitable Worlds Observatory was identified by 2020's Decadal Survey. That actually happened in 2021-22. And the Habitable Worlds Observatory has a specific goal of observing 25 Earth twins. That is, Earth-like planets around sun-like stars. So they're really going to go out there and try to find an Earth. And so what all of this means is that in your lifetime, you will come closer than ever before to knowing, is there other life in the universe? And not only do I think this, but some of the biggest names in science do. Thank you.
Awesome. Thank you so much, Megan. That was awesome. Our next speaker is Abby Elerding, who is working to identify and isolate neuronal stem cells that guide behavior in health and disease. Her research focuses on understanding how chemicals in the brain play a role in motivation and learning. These findings could pave the way for new treatments for disorders associated with abnormal dopamine activity. So please welcome Abby.
So hi everyone. Today I'll be talking about the science of motivation, exploring the brain's reward system. So imagine you wake up. It's a bright and sunny morning. You might find yourself motivated and ready to start the day. To start your day, you might choose to enjoy a nice cup of coffee, hang out with your friends, perhaps go out for a nice walk, or enjoy a nice meal. Regardless of the activity you chose, you should hopefully find this experience rewarding and find yourself motivated to do it again in the future. And this motivation is a fundamental part of life. It affects how we interact with the world and with each other, and even how we start our day. We need motivation to pursue our goals, or avoid something potentially harmful. And this is true across species. All animals share motivation to obtain their basic needs, like food, water, sex, and social interaction. And motivation not only influences our pursuit of goals and desires, but it also has significant implications for our health and well-being. And there are a number of conditions that can affect our ability to feel motivated. If you are someone struggling with depression, this condition is often associated with a lack in motivation and diminished pleasure in previously enjoyable activities. On the other hand, conditions like addiction involve a dysregulation of motivation, where individuals might show persistent and compulsive behaviors to seek and consume drugs despite negative consequences. And both disorders have significant impacts on individuals, families, and our society. And while there are treatments available for these conditions, there is a need for better and more effective treatments. And to do that, we need to understand the science behind motivation. As a neuroscientist, I'm interested in how our brains control motivation. By investigating this, I hope to uncover the insights into the complex relationship between the brain and behavior, and shed light on how dysfunction in this circuit might contribute to the development of these disorders. And I'm pretty early on in my research, so for today I'll be giving a historical perspective on this and what I hope to find out through my research. First, let's look at our brain, this large organ that allows us to think, feel, and experience the world around us. And to understand what's happening when we experience something motivating, we need to take a closer look inside. So this illustration is showing our brain if you were to look at half of it from the side. So now I want to focus your attention on this one part of our brain situated deep in the middle of it. what we can call our midbrain. And this is my favorite brain area because I study it. But the midbrain sits atop this area known as the brainstem, which connects your brain to your spinal cord, which all together make up your central nervous system. Now let's get even closer. Our brains are made up of cells that we call neurons. And these neurons make connections with one another and can send chemical messages. These chemical messages are extremely important. And different neurons all across the brain can send all sorts of messages to each other. Here's what this chemical messenger system looks like. And think of it like passing letters to one another. Here we have this first neuron, simply labeled neuron 1, that has all these packages of messenger chemicals that can be released close to neuron 2. These chemicals can then bind at these receptors, which we can call or think of as mailboxes. When this chemical binds at this receptor, this can induce a change in neuron 2 to cause it to become active, or it can tell the neuron to become less active. And this process is happening all the time in our minds. Now I want to introduce a chemical messenger that you might have heard of before, and that is dopamine. You might have heard of dopamine referred to as the feel-good or pleasure chemical because it's released when we experience something rewarding. But it's actually more complicated than that. Dopamine is not responsible for pleasure, and I'll explain why. In the 1970s, scientists had reached an agreement that dopamine plays an important role in how our brain processes rewards. And the thought at the time was that this role was in producing sensations of pleasure. And a lot of this idea can be contributed to a researcher by the name Dr. Roy Wise. And Dr. Wise worked with rats to study the brain and behavior. And even though the rat brain is a lot smaller than the human brain, they are remarkably similar in structure and function. So with these rats, he taught them to press a lever to earn a sugary reward. And these rats can learn this task really well, earning hundreds of rewards at a time. He then repeated this experiment with rats with depleted levels of dopamine. He found that these rats would not press the lever to earn sugary rewards. And Dr. Wise concluded from this that dopamine likely played a key role in pleasure because these rats had no interest in pressing the lever or even in eating the treat, and that a lack of dopamine could result in reduced enjoyment in life. And this idea was also supported by the finding that people with clinical depression tend to have lower levels of dopamine in their brain. And a few years later, another researcher entered the scene. Dr. Barrage studied the facial expressions of pleasure in rats, such as licking their lips after getting a sweet food, or opening their mouths in disgust when they encounter something bitter. When he gave rats that lacked the ability to make dopamine a sweet food, he found that they still displayed normal responses when given a sweet or bitter food, yet they rarely moved or ate on their own. He then repeated the experiment in rats with heightened dopamine levels. The rats did not show an increase in their facial expressions, but they consumed the food far more eagerly than normal. This led him to conclude that instead of producing pleasure, dopamine seemed to drive desire or motivation to consume the food. So now that we have kind of an idea of what dopamine is actually doing, let's get back to our brain. Much of our dopamine is produced in the midbrain that I mentioned earlier. From here, it is distributed all across the brain. All these areas where dopamine chemicals are being released, from neurons in the midbrain. And I'm not going to list out all of these areas by name, but one important area is called the striatum. The striatum is important for planning out actions and decisions. Dopamine released in the striatum is important for linking our motivations to actions, like choosing to study for a test because you want a good grade. Dopamine is also released in other areas of our brain, like the cortex, which is important for planning decisions to actions. So dopamine released in this area is important, like linking our decisions to actions, like I just said, like deciding to go to a quiet coffee shop to achieve the best environment to study in. And all of these areas that dopamine connects to make up our reward circuit. At its core, dopamine release in the reward circuit is important for driving our ability to seek out rewarding experiences. And in the context of addiction, the reward circuit is greatly impacted by drugs. In the late 1990s, a group of researchers looked at the activity of the brain of human volunteers with previous cocaine experience. In this experiment, volunteers were given an infusion of cocaine right before imaging their brain activity with an MRI machine. These researchers saw an increase in areas of the brain that were involved in the reward circuit. These areas that are important for driving motivation to obtain rewards. And since drugs can artificially increase the activity of these brain areas, this idea was that this could drive motivation to seek out drugs again in the future for some individuals. And studies like this clued scientists into the role of the reward circuit in addiction. But we still don't understand fully how. We know a major brain chemical involved in this is dopamine, and dopamine release is important for motivation. And now that dopamine is recognized as a key player in the reward circuit and the development of conditions like addiction, it's not the sole character in this system. There are numerous factors affecting our brain's reward circuit and dopamine itself. And to better understand how conditions like addiction can arise, as scientists, we need to get a better understanding of who and what these other characters are. And this led me to dig deeper into my research question of how is motivation encoded by our brain, and ask, what other brain chemicals interact with dopamine to guide motivation? So my research takes a closer look at chemical messengers in the midbrain. So far, I have told you that the midbrain consists of dopamine neurons, and by that I mean neurons that release dopamine. The midbrain contains another type of neuron called a GABA neuron, named that because it releases GABA. And GABA is another brain chemical that's an important regulator of the dopamine system. And together we can think of them as a gas and brake pedal in the car that is our brain. In this case, dopamine is like the accelerator, motivating us to perform specific actions, and GABA is a brake pedal in the car, telling the brain to stop its activity. If GABA is more active, it will slow down the activity of the brain. And this is what this process looks like. Here we have our GABA neuron next to our dopamine neuron. And this GABA neuron is releasing GABA, and this GABA is binding at receptors on the dopamine neuron, which in turn turns down the activity of the dopamine neuron. And we call this process inhibition. So under normal conditions, GABA controls dopamine and controls the amount of dopamine being released in the brain at a time. However, drugs like opioids can directly affect this system. They can directly bind to GABA to decrease its activity, thus in turn causing an increase in dopamine activity. If we go back to our car analogy, we can think of normal conditions as the gas and brake pedal, with GABA providing a necessary brake on the activity of dopamine neurons. Whereas in a drug condition, opioids are removing the brake, or removing GABA, thus causing increased acceleration of dopamine. So we have unchecked acceleration and excessive motivation to possibly obtain these drugs. So with this knowledge that GABA is closely connected to dopamine and impacted by drugs that have a potential for misuse, we still don't know the precise role of GABA in driving motivation. In my own research, I want to find out what that role is, and to do that, I work with mice. Decades of research and advancement in medicine can be directly tied to these small rodents and other model systems like worms, flies, and the fruit fly. Mice have simpler nervous systems compared to humans, but they have remarkably similar structure and function to us, even though it's less complex. And they also allow us to ask more precise questions about their function that we cannot do in a human. And similar to Dr. Wise's research, I work with mice to study motivation, and by training them to perform a simple action to earn a sugary treat. Instead of pulling a lever, I can teach mice to spin a Lego wheel to earn a reward. And we can measure motivation with this simple behavioral task by testing how many times is the animal willing to spin the wheel to earn a single reward. And the more motivated the animal, the more times it's willing to spin the wheel to earn one treat. We can also test their motivation by changing the value of the reward itself. Now, instead of earning a small reward, the cookie is twice as large and twice as sweet. Will the mouse perform more wheel turns to earn the reward? In other words, will it work harder than before? From what we've seen, yes, They definitely value higher valued rewards and will work harder to obtain them. But I still want to know, what is the activity of GABA neurons during this behavior? And we can look at the brain activity during this behavior task using a technique called fluorescent imaging. To explain this quickly, in my lab, we have mice that are genetically engineered to express a fluorescent protein within their brain. And think of it like attaching a tiny glow stick to a single neuron. This way, we can visualize these tiny glow sticks or individual neurons with these glow sticks or fluorescent proteins. And here's what that looks like. This is just a quick snapshot I took of a collection of GABA neurons within the brain. It might not look like much to you, but they are neurons. I've labeled some for you here in red. You might notice that some of them are white and some of them are more of a dim gray. And we can get a readout of neural activity by this difference in fluorescence. The brighter the neuron, the more active it is. Now I'm going to show you a quick video that I took of the same collection of neurons. It's a really short video, so you have to look fast. So even though that video is only 7 seconds, I hope you're able to see some of the flickering that I'm talking about. That these GABA neurons are becoming brighter and then going back to dim. And that's how we get a readout of this neuronal activity. These GABA neurons are active and are firing. And by firing, I mean they're releasing GABA chemicals. And what's great about this video is this was taken in real time from a awake and a behaving mouse. And while I only have one video to show you, with this technique, we hope to learn first, when are GABA neurons active during motivated behavior or during motivation? And since we're able to measure the activity of GABA neurons within the midbrain while a mouse is awake and performing these wheel turns to earn rewards, this allows us to answer at what points during this behavior are GABA neurons active? And down the road, how are GABA neurons regulating the activity of dopamine during motivation? And finally, how do drugs such as opioids change this interaction between GABA and dopamine to drive excessive motivation? If I lost you at any point during my talk, I hope I can leave you with these lasting points. That dopamine is an important chemical for motivation, and that GABA is an important brake on the dopamine system. And understanding the relationship between GABA and dopamine in the context of motivation could help pave the way for future treatments for conditions like addiction and depression. And with that, I want to thanks everyone for listening, and special thanks to the Engage group for putting this on.
I don't know how to work the computer, but Megan won't be talking again. Our final speaker of the night is Emma Scalisi, whose research examines the relationship between small-scale commercial fishers and fisheries management agencies in Alaska. She wants to know how fish and people can benefit from listening to both scientists and fishermen. So please welcome Emma.
Awesome. So I am really excited to be here and talk with you all about the many different experts in the world of fish and how all of their different forms of knowledge can be used to inform sustainable fishing. But before we jump into all of that, I want to talk to you a little bit about dolphins. And specifically, I want to talk to you about how obsessed with dolphins I was as a child. I had so many dolphin t-shirts that I wore constantly. I covered everything I owned in dolphin stickers. I read too many books where the main character was best friends with dolphin. And I don't know if any of you know what horse girls are, but I was definitely a dolphin girl. And this love of dolphins had a strong enough influence on me that I decided I wanted to be a marine biologist when I grew up. I wanted to be somebody who studied the ocean. So how did I end up here tonight where I'll be talking to you about fun things like politics and management decisions when I really just wanted to be out on a boat becoming best friends with the dolphins? Well, it turns out as I got older, I realized that what I was most interested in was learning how we could form more sustainable relationships between humans and the ocean. And when we're thinking about sustainability, there are really broadly sort of a couple of different ways that we can think about these concepts. And the first views humans as separate from nature. This is sort of this conservation view, and it's a relatively recent Western notion of how humans interact with nature. And the idea is that minimizing these interactions is what is best for nature. And then another viewpoint is to look at humans as a part of nature. And so this doesn't view human interactions with nature as inherently harmful, and instead it sees that we can have interactions that are beneficial for both humans and nature at the same time. And it's this viewpoint that really resonates with me. And if we're thinking about this in the context of the ocean, it can look a lot of different ways. And it can include things like fishing and recreation and tourism, culture, using the ocean as a source of power. And while a lot of typical conservation strategies work to manage the ocean by separating people from it, this isn't always necessarily good for people or the ocean. And so that brings us to this sort of broad goal of my research, which is to figure out how we can meet the needs of both humans and the ocean at the same time. And specifically, I think about that in the context of fishing. And so I'm probably going to use the term fishery too many times during this presentation, so I just want to make sure we're sort of all on the same page when I use that term. And so a lot of times when people hear the word fishery, they might think of the people who are catching the fish, or they might think of the fish themselves. And in reality, it's both of those things. So it's a system of both the people catching the fish and the fish themselves. And there are a few different sort of categories of fisheries that exist. The first is subsistence or artisanal fisheries. These are usually smaller in scale, and people are using them for an important source of food, and these are also often culturally significant. The second is recreational fisheries. So if you've ever been out like fishing for salmon in Puget Sound, it's fishing that's done for sport or for fun, and maybe you'll also eat the fish. And then the last group are commercial fisheries. These are the fisheries where someone is catching and then selling the fish. And within commercial fisheries, there's this pretty broad scale of what they can look like, all the way from these like really big boats with dozens of people on them. They can be out on the ocean for weeks or even months at a time, all the way to these tiny little 1 or 2 person boats staying pretty close to shore. And these are the specific type of fisheries that I study, and we call them small boat commercial fisheries. And unfortunately, when we think about all of these different types of fisheries, a lot of them around the world are struggling right now. There are a lot of different pressures they're facing. So there's more fishing. We have a lot more people than we ever have before. And because of that, there's more demand for seafood. And there's also more advanced fishing technology, which makes it easier to catch more fish. And then on top of that, we also have a lot of environmental stressors. So pollution, warmer ocean temperatures, ocean acidification, things like that. And all of these together contribute to a higher number of unsustainable fisheries over time. And when I say unsustainable, I basically just mean that if we kept fishing at the same rate, eventually we would run out of that type of fish. And so what this graph here is showing us is a couple of things. It's basically showing us the change in unsustainable versus sustainable fisheries over time, specifically since about the 1970s till today. And we can see a couple things. The first is that this orange bar on the top, showing us unsustainable fisheries, has increased pretty significantly. But fortunately, what we can also see is that there are a fair number of fisheries in the blue there that are still quite sustainable, which is great. So if we're thinking back to that goal of supporting both humans and the ocean, what we want to work towards is minimizing this orange bar, right? Lowering the number of unsustainable fisheries and raising the number of sustainable ones. And before we jump into some solutions, why specifically do we want to keep fishing rather than just saying, hey, we're going to stop fishing and then we know we're not taking too many fish out of the water? Well, fisheries are really important for a lot of different reasons. Like we said earlier, a lot of people around the world rely on them as an important food source. They provide money and jobs for tons of people. They can be really culturally significant. And for some people, they are an important part of their identity and autonomy. There are lots of commercial fishermen who I've talked to who could definitely be making more money doing something besides fishing, but there's no way that they're changing jobs. And there are tons of different people who are working to keep fisheries sustainable for all of these different reasons and plenty more. And so tonight, I want to talk specifically about 3 different types of experts in the world of fisheries. First, we have our fishermen. They're the ones who are catching and then selling the fish. Next, we have our scientists. These are the people who are studying the fish, trying to figure out how many fish there are, where they are, things like that. And then lastly, we have the managers. So these are the people who are making the rules that the fishermen have to follow, like where they're allowed to fish, how many fish they can catch, things like that. And so how does this all work? Before I fully get into that, I want to make a quick note that I work specifically with fisheries in the United States. And so while some of this is applicable in other places outside of the US, this is specifically where I'm talking about. So first, we have our scientists, and they are going out in the ocean. They are doing surveys to count how many fish there are, where the fish are. They're getting information from the fishermen about how many fish are already being caught. And then they're putting these into really fancy models to predict how many fish there will be in the future. And they give this information to, I don't know why this slide got funky. To management. And so management, right, are the people who are making the rules for fishermen. So they might put into place limitations on how many fish you can catch, how big a fish has to be for you to keep it, when you're allowed to fish, things like that. And if we think back to earlier, how we saw an increase in the number of unsustainable fisheries, more of these management tools are being used. There's more limitations on fishing being put in place. And often, hopefully, this is good for the fish, right? It's making sure that their populations stay healthy. But it can also have a lot of negative consequences for fishermen and fishing communities. And so you might have noticed that I said we had 3 types of experts in fisheries, but I'm only talking about 2 right now. And you would be completely right. I don't know if any of you have ever worked in a job and had a boss or a manager who was constantly telling you to do things in a way that just really did not make sense to you. Because I know that I have, and it's super frustrating because the managers probably had a good reason for asking you to do things in a certain way, but I'm sure you also had a great reason for why that just didn't make any sense. And this is often the dynamic we see in fisheries, because while scientists and managers have a lot of important knowledge and information, they have these records that are kept over time, really meticulous, fancy spreadsheets that are being recorded. They're missing some really key knowledge from the fishermen. And so our current system of management doesn't include all knowledge. We're lacking information that fishermen might have on generational knowledge that's been passed down, local ecological knowledge that they've observed when they've been out on the ocean, and really importantly, information on what fishermen specifically want and need to continue using this as their livelihood. And this is a really big problem also because it means that scientists and managers aren't including all of the information in their decisions. So fishermen, as you might guess, spend tons of time out on the ocean, and they might see things that scientists are completely missing. So there might be fewer fish than what the scientists are seeing, there might be more fish, or there might even be totally different types of fish out there than what the scientists saw when they were out there for just a couple of days. And so because this isn't getting communicated to management, they're not making the best possible decisions. And this isn't meeting that goal of being a best practice for the ocean or for fishermen. And so how do we solve this? Something that I find really frustrating is that when we look at those unsustainable fish populations, a lot of those declines in fisheries have been directly attributed to a lack of communication between fishermen and management. And I find that frustrating because there's something specific that could be done. But at the same time, I think this is really hopeful because it gives us a concrete change that can be made that would be really beneficial. And so right now, we have sort of this one-way flow of knowledge from management and scientists to the fishermen. But really, what we want to do is just add that flow of knowledge back from the fishermen to scientists and managers. So if you think back to that annoying job you had, it would be giving you the opportunity to go to your boss and say, hey, this is why I'm doing it this way, and this is why I think it makes sense. This is what you're missing. And so the research that I specifically do is in Juneau, Alaska, and we talk to fishermen and scientists and management to try to get a sense of two things. And the first is, what ways are fishermen already sharing their knowledge with management? And the second is management helping to support fishermen in this? And what does that look like? So my work is specifically based in Juneau for a couple of reasons. In Alaska, as I'm sure a lot of you know, there's tons of fishing happening already. But Juneau is kind of unique in that it also has a lot of management offices there. And so compared to other more rural places, there's more opportunities potentially for this connection and collaboration between scientists and managers. And then the second reason is that the advisor who I worked with already has some personal relationships and connections to Juneau. And I think this is really important when doing research in a community to have this connection, because that way you're not just coming in, asking a bunch of questions, and then disappearing. And so my actual research looks a little bit different than a lot of other scientists, because the main way that I do research is by talking to people. So we did interviews with commercial fishermen and managers and scientists to try to figure out sort of what's happening right now and how could changes be made so that fishermen's expertise are included in management decisions. And I was actually kind of nervous going into these interviews. I don't know if any of you know any commercial fishermen, but they are not people who like to have their time wasted. They are quite busy. A few of the interviews that we did happened while fishermen were like literally fixing their engines or cleaning their boat, things like that. But fortunately, when you go to someone and you say, hey, do you want to complain about your boss? And also it's for science. They're pretty happy to talk to you, which was great. So what did we learn? Broadly, it's really complicated. But I think that it was summed up really well by this specific quote from a small boat commercial fisherman who said, in all politics, it's the same thing. Whoever has the most money and can buy the most time is going to get the best representation. And I imagine this sounds familiar to a lot of you. This is not something that's unique to commercial fisheries by any means. And if we think back to earlier about sort of those different types of commercial fisheries, these really big boats that are out on the water for weeks or months at a time, they are making a lot more money than these smaller 1 or 2 person boats. And this money can be used for a lot of important things, like maybe these fishermen who are making more money don't have to take a second job, so they have more time to talk to management and share what they need. Maybe they can even take these managers out for drinks and dinner and get them to listen to them a little bit. And if they're in a high enough earning fishery, fishermen will sometimes come together and pool their money to donate it to administrations. And then that administration feels an obligation to listen to them and to their needs. But if you're in this tiny little boat, right, not making a ton of money, you're not able to do that. So some voices in our fisheries are being heard a lot louder than others. So kind of just to like sum it up, right? If you make more money, you directly are more able to make these important social connections, and those social connections lead to policy decisions. And often, those are policy decisions that are being made in favor of the people who already have more money. So, if our goal is to hear all of these voices, including the voices of these small boat commercial fishermen, basically the biggest barrier to that is politics. And when we talked to fishermen and we said, hey, it seems like talking to management and sharing your needs and concern and knowledge is really important, why do you think that isn't happening more? What one person in one of these smaller fisheries told us is that their time sharing knowledge with management would be better invested at this point in my life taking care of me, because I don't think I'm going to have any impact. And again, this is really not sort of the ideal situation. There are clearly a lot of barriers to this communication. And we heard other barriers too when talking to other fishermen. We heard that there's just a general lack of trust between fishermen and management, that fishermen said, we don't really want to tell them things because we don't know or we don't trust that they'll do what's in our best interest. We heard that there was just too much bureaucracy. Commercial fishermen don't tend to get into that job because they love sitting in a bunch of meetings, but that's often what talking with management looks like. And lastly, just thinking about social norms. We heard fishermen say, I would talk to management, I would sort of tell them more information, but I don't want other fishermen knowing that I did that, because that would look like I was, quote, on the side of management, and that would look really bad for me. But fortunately, we also heard a lot of really hopeful things from these fishermen as well. We heard fishermen say that they spent a lot of time advocating. Even fishermen who had to take second jobs to make extra money often took second jobs in an advocacy role to try to promote ocean sustainability or fishing sustainability. We also heard that more informal communication is really common. So fishermen sort of slowly getting to know management over time, building these relationships with them, and eventually feeling comfortable enough to share their knowledge with them. And one specific example we heard that I think is really cool is a manager telling us that over time he had gotten to know this fisherman, and there was a specific species that they were worried about, and they were like trying to avoid being caught. And the fisherman said, hey, I'm out on the water. I know that in this one area, I'm catching a lot of that species. If I were you, I would close that area. And the management said, okay, great, let's close that area. And so there is some of this collaborative management happening. And then lastly, we were hearing fishermen tell us all of the ways that they're taking care of their fisheries regardless of what management requires. So fishermen saying, I know that if I fish over here, I'm not going to catch the species that I want, or I'm going to catch bycatch. So I'm going to avoid that area even if I don't have to. Or other fishermen saying, I know that the biggest fish in the ecosystem are the ones that will produce the most offspring. So even though I could keep this really big fish, I'm going to release it back into the ocean, hoping that it produces more fish. And I think things like this were really hopeful to see how fishermen are sort of already working to meet this goal of both their own needs and the ocean at the same time. And so our hope is that when we're understanding all of the barriers to this communication, as well as what's already in place and already working, that we can make some changes that will be beneficial for both the fishermen and the ocean. And if you're feeling really stoked on fisheries management after this, my really good friend Lucy Bowser will be right here next week talking about fisheries management and conservation on an international level. So I definitely encourage you to come hear her talk. And thank you so much.
Okay, is this working? It's really hard to tell if your own voice is amplified. Okay, cool. If anybody has any questions, please come up to our mic and ask them. We want to hear them. We want to answer all of your questions.
Hello, I might not want to know the answer, but how do you get the image of the brain of the mouse?
There are a lot of ways we can do that. I keep this vague, but we use like tiny microscopes essentially to capture images of the brain. But yeah, there are a variety of ways you can do it. Yeah, I'll go too into detail, but it's tiny microscopes. I'll leave you with that.
So, what does oxygen look like?
That's a good question. You can see oxygen now. So with your eyes, it doesn't look like much, but as an astronomer, to detect oxygen, what we do is a type of science called spectroscopy. So we wait until a planet is in front of its star or its sun, and then we have light passing through the atmosphere of that planet from the star, and then we can capture it with our telescope. As it turns out, light is not all the same thing. You have different colors of light, is what you can think of it, like red, blue, green, all of those kinds of light. We can actually tell the difference between based on how much energy they carry. And so if we look at the light coming through the atmosphere, some of it is going to get absorbed by the oxygen. So you might remember from like a chemistry class in grade school, different transitions in electrons will need different amounts of energy, and that removes different amounts of light. And so if we look for what types or what colors of light are missing, we can infer what gases are present in an atmosphere.
We like chemistry. Okay, it's chemistry. Any other questions? Feel free to come up. Sure.
I'll use my loud voice. So if you're looking at exoplanets going in front of the stars, when we look out there, do we have any way of finding exoplanets that are going this way from our point of view, or do they have to be sort of in the plane with us?
Also a very good question. So we are by far and large biased towards planets that pass in front of their star. However, alternate detection methods are very common and are becoming more popular. The number one method for detecting planets that maybe don't pass directly in front of their star is called radial velocity. So gravity, for those of you, of course, you know gravity holds you to the planet. Gravity is what holds the Earth around the sun. But for every force, there is an equal and opposite reaction. And so as the sun holds the Earth in its orbit, Earth also pulls on the sun. And so it still has to be that the solar system that we're looking at is edge-on in some way, but even if the planet doesn't pass directly in front of its star, we can still sometimes see that it's tugging on its star, and that can help detect the planet is there. There are other methods as well called gravitational lensing is one. That is a very tricky one that I won't get into, and stuff of that nature. But yeah, we are absolutely biased, and this type of spectroscopy is very hard to do if you don't pass in front of your star. There are a technique called direct imaging, which can be accomplished even if it doesn't pass in front of its star, and that's going to be the type of observations that the upcoming Habitable Worlds Observatory will be conducting.
Go ahead. Oh, sorry.
I'm curious on the topic of neuroscience, if there's a difference in the dopamine production or the GABA production before the person, say the addict, becomes addicted to drugs, and if that could be identified early, and then some different kinds of treatments or conditions could be set up to prevent that person from being an addict.
That's a really good question. A number of drugs affect the dopamine system in a variety of ways. It's not only through GABA like opioids do. Mm-hmm. Cocaine, for example, affects dopamine transport. Yeah, that's a good question. How do I answer that?
Is the question like, are people maybe predispositioned?
Yeah.
Yes.
Yeah, that's definitely the case. I think like genetics play a strong role in how if someone is more predisposed, whatever the word is, to develop an addiction. And that's not really what I study, is like the genetic component. Obviously, I focus more on how GABA controls dopamine, and specifically in motivation. Motivation is not the only aspect that drives addiction also. So I think that's a really good question, and I just focus on the motivational component of it and how drugs affect GABA and dopamine and that relationship. But yeah, other drugs can affect dopamine in different ways, and also different neurotransmitters or brain chemicals that I didn't talk about today, such as like serotonin, and also like endogenous or opioids that are produced within your brain that can be affected by drugs.
Or even different parenting styles that would exacerbate the production of dopamine
definitely.
without.
Well, definitely like societal factors can affect addiction. Yes.
Thank you.
Yeah.
Okay, awesome. Yes, nature and nurture are both very important.
Yes.
That's what we like to say. We had a question in the back.
Yeah, and I'll use my live voice. As far as like fisheries, I know in your presentation you showed that graph of sustainable and unsustainable fisheries. Yeah. My understanding is that's a global statistic because it was coming from the FAO. Yeah. What about fisheries like specific to the Pacific Northwest and like the Bering Sea? Like, should we be optimistic or pessimistic about those fisheries?
Yeah, I mean that is a big question. If we're looking specifically at like the Pacific Northwest and Alaska, I know we hear a lot of doom and gloom statistics around fisheries, but specifically around here, like a majority of our fisheries are doing really great, which is awesome. It just happens to be that some of these sort of like really almost like charismatic or culturally important species like salmon, or if you've heard of like the Bering Sea crab fishery closure, some of these sort of big ticket species are struggling right now. But I mean, I don't think I could be in this work if I didn't still have hope, right? And so I think there are certainly species that are struggling, but in the Pacific Northwest at large, like, we're doing pretty good.
Awesome. You mentioned salmon, and it made me think of another awesome student in our class who is giving a talk on May 22nd. I think it's the last day, Tessa. She studies the declining salmon population in Lake Washington. So if you're curious about salmon, you should definitely come and listen to that talk as well. Does anybody else have any questions? Go ahead.
I'll keep going. With the sustainable fisheries graph, are we who don't access the international data on a regular basis, the sustainable seafood stickers in the supermarket, are they actually pretty reliable, and do they match what you're finding for sustainable fisheries?
Yeah, that's also a really great question. I would say the broad answer is I think yes. I think those are a really great starting point. And the sort of more detailed answer is I think it depends on like sort of what you personally care most about in your I don't know, shopping choices. So if you're like, oh, I want to make sure that like there's a low bycatch, or that fishermen are being paid well, or things like that. There's so much research you could dig into with it, but those stickers are a great starting point.
Anybody else? I'm going to keep asking anybody else.
She's going to start the quiz if nobody has.
Yeah, this is actually science class. I actually have another question for you, Emma, about fisheries. In terms of like the interactions that you've had with the fishermen in Alaska, do you think that the findings that you guys have determined, even from this very small area, will, I guess, kind of are able to be expanded to other areas, or is it very like personalized by location and like the interactions that people have in their work?
I think that's also a really good question, and something that people in this field sort of think a lot about is what's the balance between something being like really specific versus something that you could apply to any community. And so I think a lot of it is quite specific to these people and their specific set of circumstances, but I still think there are lessons that we can take away from it that are broadly applicable. So like, I bet in tons of fisheries all over the world, you'll see that like, if there's more money, you can make more friends and you're at an advantage, right? So.
Anybody come up with a new question while I ask that one?
In the GABA movie, why was it pulsing?
Why was it pulsing?
Yeah.
That was how we get a readout of our brain activity, or of the GABA neuron activity because of by that pulsing, like the flickering you see of like becoming more white versus like a gray. The more white it is, like the more active it is. So there's just like change in fluorescence, essentially, of the proteins that are expressed in those neurons.
Does it sync with brain waves?
Yes, yes. Brain activity, yes.
But why does the same one turn on and off and on and off again instead of just firing once?
Because our neurons are constantly firing in our brains, just all the time. So that's why you see like that pulsing happening. It's not really a case of where like a neuron fires and it just stops. It's usually just happening all the time. So that's why you're seeing that flickering. And then there's some you might have saw in that video that didn't flicker at all. They probably just might have died, and that's why they didn't. But luckily, you don't need all your GABA neurons.
Anybody else? Otherwise, I have another question for Megan. Sorry, we're putting you back on the spot. It's been a minute since we talked about outer space, so I am curious what you can share about, I guess, kind of your work in modeling this like water loss. What are you trying to find there, and just kind of like, what's the goal of that?
I don't know if this microphone works. But
I think you have a loud voice, so you're good.
that's true. Now I have two. I'm just kidding. If you want this. So in my work, yes, the process I talked about is the process that I model, but I specifically apply it to a target of interest called TRAPPIST-1. You might have heard of TRAPPIST-1. It's very popular right now, and it has been in a very long time, and for good reason. But it has 3 planets that are in what we call the potentially habitable zone. So the right distance from the star that liquid water could be sustained at the surface. Now the issue is that TRAPPIST-1 is not like our sun. It's a much smaller and colder star, and it also has a period of activity that our sun does not have called the pre-main sequence phase. And during this phase, the X-ray and ultraviolet radiation from TRAPPIST-1 can be multiple orders of magnitude greater than it is today. And the issue with that is that all of the planets in the habitable zone today used to be in the Venus zone when TRAPPIST-1 was born. And so what we're worried about is when you have lots of radiation, the process that I was talking about can occur. And so we're like, oh my God, what if these planets have lost all of their water before they ever made it to the habitable zone? Then what do we do? So I run simulations that calculate how much water you're going to lose based on changing radiation over time. And I test out different amounts of initial water contents. And I try to see, A, how much water can these planets lose, and B, how much oxygen does the process produce. And the goal is to answer 2 questions. Question number 1, is there water left over, and is it likely there will be water left over? And question number 2, is enough oxygen being produced that we can't trust it as a biosignature on these planets?
Thank you. Kevin.
Yeah, I have a question for Abby. So you're studying mice, and I'm wondering what kind of differences, or maybe not differences, there are between mice brains and human brains.
Yeah, there are some differences. Mouse brains, well, visually, they're a lot smaller than our brains. They also have a lot smaller of a cortex than we do. Cortex obviously is involved in higher order processing, like thinking, decision making. So mice are making less thinking, less decisions. So that's like the biggest difference I would say is like the cortex is a lot smaller than ours, and like that's the outer area of your brain. And yeah, I would say like that's the biggest thing I could think of right now is the cortex, just the size difference. They have also a lot fewer neurons than we do, and less complexity in terms of like structure and function, but the basic function remain the same. So.
I'll do an outer space question. I would speculate that an inhabited planet has much greater seasonal variations in its gas than an uninhabited planet. Can you look at these planets at different times in their orbits and still get and what is that showing you? Or,
that's a very good intuition, actually, because we do have a whole area of research into seasonal biosignatures, aptly named. And
in general, some biosignatures aren't really going to change that much. Like if you look at Earth in the winter, you're still going to see oxygen, and if you look at Earth in the summer, you're still going to see oxygen. But some biosignatures do change over time, and seeing changing conditions over time in and of itself can be a biosignature in some cases. So for example, plants. I didn't talk about this before, and I won't get into the specifics, but one thing that we look for is something called the vegetation red edge. And that has to do with the light that gets reflected off of a planet that's covered in vegetation. And so in the winter, maybe you're not going to have as much vegetation, and so that biosignature isn't going to be as strong. So that's an example of one that would be affected by the position in the orbit. Other things like gases produced can change over time, and seeing that you have gases that are waning and ebbing over time can be a biosignature, showing that life is, flourishing maybe in the summer and hibernating in the winter. But those are hard to detect, and we probably won't detect those at first.
Cool. Okay, so apparently I'm losing my voice, even though I haven't been talking. We're nearing the end of our Q&A session, I believe. Any last questions for our lovely scientists? Yes.
I have one for everybody. What's your favorite part of the research? Like, that you do? What do you enjoy the most?
I guess I'll start. It's the hardest question. Maybe this is really basic, but every time you get an unexpected result, and then I show my advisor, and he's like, we shouldn't be seeing that. And then I'm like, well, I guess we're seeing it. I think that's the most exciting thing, is just when you find something unexpected, and then you realize, like, maybe we were thinking about this wrong the whole time, or maybe my experiment is completely wrong. And just that unknown is, like, the most exciting part to me, I think. And also seeing the excitement in other people's faces when I tell them.
My science answer is that, that's a really good answer. But my science answer is that I want to know if aliens are real. And I get really excited when we get new data and I get to know about it before the public does. So that feels pretty cool. And then I get to interpret it, and that feels even cooler. And then my life answer to that question is all of my work can be done on the computer, and a lot of times it's done from my bed.
I think I touched, I don't know if this is working.
Here.
I think I touched on this a little bit in my presentation, but just getting to talk, I think specifically to people like commercial fishermen who are like, I don't know, I'm a grad student in the city. Like, we live extremely different lives, and then talking to them and being like, wow, we are excited about the same things and share all of these values about wanting to support the ocean and wanting to support fishing communities. I think those connections are really exciting. Yeah.
Awesome. My favorite part of my job was that I got to work with all of you. So, any other last questions? Okay, I'm going to wrap it up then. I think I have a nice little piece of paper that tells me what I have to say next. Okay, yes, thank you guys all so much for joining us tonight. If you enjoyed this, you should come back. There are 4 more nights like this, and the next one is on May 3rd. It also starts at 5:30. It is here, but in a different room, I believe, in the reading room. All of the information is on the Town Hall website, so you should check it out. We have 3 different speakers talking about 3 other completely different things. We have the follow-up to the fisheries talk from Lucy, who's actually here in the back. Not to put you on the spot, sorry. So we'd love to see you back here. Additionally, if you were kind of curious about Engage, you're welcome to come chat with me after this. I'm happy to share any information. You can also visit our website, which is engage-science.space, or we also have social media. So you can follow us on Instagram or Twitter at Engage Science. And also, if you're a graduate student and you're here and you want to learn more about science communication, then you should consider applying for this course next year. Applications open in November for the winter quarter class offering. So it will be winter 2024 for the class. But thank you guys all again so much for coming. And we really appreciate, getting to talk to the public. So thank you.